shows you what data is available, how to approach requesting these data.
Thanks, Cindy. Hi, I'm Shirley Johnson, and I'll be joined by Rachel Wilson in a moment, and Toby Bedhauser will be talking after us. We're going to talk about the ERT approach to biomarkers. As you just heard from Dr. Carlson, it's really critical to have biomarkers if you want an accurate diagnosis. The medical record is fraught with diagnoses that are clinically based, and they're going to be not accurate about 20% of the time, 20 to 30% of the time. I wanted to start with this title because it's provocative, and it also tells you the insights that we are gaining from biomarkers, that there's the disease itself, which is biological diagnosis based on proteinopathies. And then there's the clinical syndrome that emerges, which we usually think of as Alzheimer's, but in fact, that's a late stage of the disease. As you've already heard, the pathology can now be encapsulated with in vivo imaging, so we don't have to wait till a person passes away to know if they have amyloid or tau in their brain. And this is a good example of that. Two people with mild cognitive impairment diagnosed with 5 hours of cognitive testing. One of these people, the one on the left, is different than the one on the right. This person on the left, their amyloid scan on the upper left is very positive, and their tau scan in the lower left is very positive. And you can see that from the back of the room. And this is the value of biomarkers. Two people with presumed mild cognitive impairment have different etiologies, and you won't know that until you look at the biomarkers. This is a single case that we studied over time for age 66 to 71. And each of those rows is one of the exams looking at the brain from different views. The main thing I want you to get from this is that the disease progresses gradually, but you can see this over a 5-year period. You can see this grow from a very small focal area to covering most of the posterior parietal and temporal parts of the brain. When we put all that kind of longitudinal change together, this is looking at amyloid PET scans now, we see these kinds of patterns. This is data from our center, and I'll try to do this with the mouse here. This is just the raw unadjusted data here on the left side. You can see these trajectories in amyloid over time are fairly consistent from person to person. The people in red are amyloid positive, judged after the fact based on a threshold that we derive, and the ones in black are amyloid negative. These rates of change are so characteristic that you can fit a function to this and get an idea of when we may have crossed that threshold to become amyloid positive. This gives us a way of anchoring the disease to a starting event, if you will, a starting point that tells us when the disease began. That's going to help us study resilience and susceptibility and so forth. It's not just our data that show this. Our data is there in the upper left. Washington's data shows this. Mayo's data shows this. The Baltimore study on aging and the multi-site ADDM study all show this kind of pattern. This is data from Toby Bedhauser's important work. And this also from Toby Bedhauser's paper showing that you can use this kind of longitudinal characterization to identify the stage of onset, which is here. And we've done this by different ApoE genotypes. So let's just look at the green one in the middle, ApoE33. The onset age for amyloid for the disease itself, not the symptom, but the disease itself, has a 50-year span of time. So this is really important that we anchor our investigations to the biology and not necessarily to the later clinical syndrome. This slide is just to show that the longer you've had amyloid in your brain, the more likely you are to also have the other protein involved in Alzheimer's disease, which is tau. And finally, I want to show you this one, which is the symptoms on the y-axis, which we use this instrument called the CDR, clinical dementia rating. We've got their amyloid time on the x-axis, and you can see that the average time to reach dementia, which is roughly around 5 on that y-axis, takes about 21 years or so. But there's a ton of heterogeneity here. And some people are ahead of the curve, they're reaching symptoms sooner. Some people are resistant to reaching symptoms. And we need to study both of those groups and not rely on simple averages. We need to know why some people are susceptible. Do they have co-pathology like alpha-synuclein GDP or vascular disease like Dr. Carlson just mentioned? And why are these people who are thing resistant, the small subgroup down here, why aren't they progressing? We're going to learn a lot from people like this. This is the mission of our brand new study that's launched across the entire EDRC system. We're calling it Clarity. The goal here is accurate, individualized biological diagnosis and staging so that we can understand cognitive impairment with more clarity than we do now. Okay, then I will now turn it over to Dr. Rachel Wilson, who will tell us more about the fluid biology.
Okay, so I'm going to start by just giving a little intro to our lab at the Biomarker Core of EdDem, and we'll be doing tours later, so hopefully some of this will look familiar when you're walking to the lab. I just want to mention that part of what we do as a core lab is work with people to decide what analyses may be useful for you when it comes to analyzing biofluids, the assays that are available, kind of what the field is showing. So if you have any questions or a project you may be interested in collecting biofluids on, please feel free to reach out. We love working with people all across campus and the country. Okay, so I'm not going to spend a lot of time on this since we talked about it quite a bit, just to say that, again, the goal of the revised criteria was to anchor the diagnosis of Alzheimer's disease to the biology rather than the syndrome, and also to show where we have validated biomarkers and where we do not yet have validated biomarkers. And I'm mostly going to focus on the core one, which are the diagnostic biomarkers. Okay, so all of these data that I'm going to show, for the most part, were from samples analyzed in our lab from our rapid EDRC cohorts. So just going back a few months after I started here with EDRC, the first commercial p-tau217 assay became available. And this was a big deal because up until that point, we could really only send samples to other research labs to have them analyzed. So there was, it was showing a lot of promise, but there wasn't a lot of robust comparisons across labs. And so our goal was to take all of our plasma, and we have over 6,000. This is 5,500 roughly, and characterize it with this new commercial p-tau217 assay. So we analyzed all those samples, and what you're seeing here is every dot is a time point, every line is a participant. So you can see there's quite a bit of longitudinal data. And in evaluating p-tau217 as a useful biomarker for ED diagnosis, of course, one of the first things we wanted to look for was how well does it agree longitudinally with our known biomarkers, and in this case, gold standard PET. So we stratified the plasma trajectories by people that were amyloid PET negative, people that were amyloid PET positive, and I think just from looking at this, you can see a pretty clear difference that people with that are amyloid positive, the p-tau concentration is on the y-axis, and their age is on the x. They're obviously showing an increase in age over time that is different from what we generally see in amyloid negative. And again, we want to make sure that the changes that we're seeing over time with p-tau217 also reflect what we would see So this is a smaller subset of the previous samples that I showed. And these are people that had a p-tau217 measurement within a year of an amyloid PET. And again, we see that the people that are amyloid negative, for the most part, they're not really changing on PET or plasma p-tau. And for amyloid positive cases that, they're increasing, the p-tau217 is increasing as the PET increases, which is, reassuring what we want to see. We also looked at the diagnostic accuracy compared to our gold standard PET. And for these ROC curves, in perfect agreement would be an Mariana curve of 1, and we get really good agreement with p-tau217 in comparing it to PET. One thing I do want to point out is that when you see these area under the curves and these predicted values and these thresholds, they are representative of the sample that you're basing it on. So in this case, we see a higher negative predictive values, higher positive predictive values when we're detecting any amyloid positive case. On the right side, this is specifically for early amyloid positivity. So these are people that are going from A negative, T negative to A positive, T negative. So they are likely not impaired. I don't know that for sure for everybody, but this is very early disease. And even though we lose some of the area in the curve in our predicted values, this is still really, really good for being able to diagnose the disease, 20 years before symptom onset. And knowing that Alzheimer's, the biology progresses relatively slowly in the preclinical phase. I think these are still really shows a lot of confidence that p-tau is reflecting what we see on the PET. The main approach that has been taken now with p-tau217, at least in the clinical workflow, is to adjust the threshold so that we have a upper threshold for people that are very likely positive. We have a lower threshold for people that are very likely negative, and then this intermediate or indeterminate group that are in the middle. And the goal of using this workflow clinically is that these people that are in the middle, they can go on for confirmatory testing, but these people that are clearly positive or clearly negative, we can spare them that, lumbar puncture or PET. The diagnostic accuracy in this upper threshold range is very high. And again, just showing that we can kind of manipulate the accuracies and the size of this intermediate range by adjusting where those upper and lower cutoffs are. I'm not going to go too much into why p-tau217 is not a good HOR2 biomarker. We do have a lot of data showing this. This is something we can talk about during the tour. But I hope that I've shown that we can have a lot of confidence in p-tau217 as a HOR1 biomarker for diagnosing Alzheimer's disease and the presence of amyloid. And a lot of the work that we have done in our lab has been a really beautiful example of the power of translational research, because we did this, the previous data I showed was far ahead of the commercial validation or the FDA validation of this test. So our data helped gain confidence across the field in this assay. And we also worked with Fujifilm, the company that had the first FDA cleared blood test for AD diagnosis. And as we've talked about quite a bit, and looking at where we're going now, we have a really good biomarker for diagnosing AD. We don't have really any biomarkers, at least, especially fluid biomarkers for diagnosing these other proteinopathies that we know commonly occur with Alzheimer's. And the problem with this is that this is likely what's contributing to a lot of that heterogeneity that we see in symptom onset and progression. So this is a data set that we ran on the new LISA Almer Argo instrument here. And it's a targeted proteomics panel, like Dr. Sana had spoke about. And it was really interesting because when we compared people that were unimpaired versus those that had NCI, p-tau217 was our top hit, as we expected, because our cohort is 80 focused. But then this phospho-TDP43 was the second highest hit. And this is a biomarker that is indicative of TDP43 pathology, things like late and some FTDs. So we looked at this a little bit further in the context of Alzheimer's. And what we saw in this panel on the right is that people that had an elevated p-tau or an elevated phospho-TDP and were amyloid positive were declining faster in their cognitive testing than those that were positive for amyloid but have lower amounts of phospho-TDP. So this does seem to be accounting for some of the heterogeneity. Of course, this is just a single study and really needs to be robustly replicated. We've seen some data coming out of that look very similar to ours. So in that note, this is where we're going. We're working on developing assays for detecting these other pathologies because we know that Alzheimer's is not the full picture here. And plasma p-tau217 is a very reliable biomarker for amyloid positivity. You know, we have to be cautious with the cut points and what people are reporting and accuracies. And to remember, with the 3-tier approach, blood biomarkers are meant to be used in conjunction with clinical evaluation and are not meant to be standalone diagnostics. I think that's it for me. Here's our whole look at our lives.
I'm not sure who's next. Oh wait.